In Chapter 14, we look at evidence of evolutionary change. In other words, what aspects of the natural world do scientists look at that serve as clear indicators that natural selection, evolution by natural selection, does occur? So one of those is shown on our screen here, and so this is an example we've talked about a number of times during the course of our class. On the left-hand side, we see the prevalence of malaria in the world, and on the right-hand side, we see the prevalence of the sickle cell allele. And so we've had a number of discussions in our class in an entire lab devoted to a discussion of the prevalence of that sickle cell allele, where it came from, what advantage it provides. And so it's, it of course would have to be the case that that allele is most common in areas of the world where malaria is also common. So going back to our bullet points here, what we see is this. The theory of evolution states that all living things have a common ancestor. This is known as the common ancestor theory. Each organism is adapted to a particular way of life, and we remind ourselves that when we use the word theory in the world of science, we're not talking about a random guess, but rather we're talking about a well-established hypothesis that's been tested over and over and over again, and each time it's passed that test, and so we have a great deal of confidence in hypotheses. The theory of evolution is a unifying theme in biology. It ties together anatomy, physiology, certainly ecology, microbiology, unquestionably molecular biology. And so all of these different fields draw on evolution, and to be, it's often said, and is certainly the case, it's very difficult to make sense of anything in the world of biology without this unifying theme of evolution. So now we make our way to several bullet points discussed by our text that serve as evidence for evolution. Number one is the fossil record. So, fossils are certainly an interesting topic. We live here in central Kentucky and are surrounded by fossils. All the limestone that we have here in Madison County, to the best of my knowledge, in any case, all the limestone that I observe in Madison County contains brachiopods. And so those brachiopods, you know those as little shells if you're not familiar with the term brachiopod. There are fossils absolutely everywhere, in other words. And as we examine those fossils, not that Madison County provides all this evidence, but worldwide as we look at these fossils, then we see that it provides an interesting picture of the progression of life as we go from the origin of prokaryotic life in waters to eventually vertebrate life on, eukaryotic, of course, vertebrate life on land. Your book has an interesting example of Tiktaalik, and which is shown here in the middle of our diagram, which is an interesting transitional fossil. And so Tiktaalik has characteristics of both the primitive fish that existed before it and coexisted with this organism, and amphibians that arose after it. And so Tiktaalik is an interesting example of what we refer to as a transitional fossil. In this case, a transition between fish and amphibians. The next piece of evidence for evolution that we examine in our text is something referred to as biogeography. And so in biogeography, what we see is the following: that as we look at the distribution of animals that are found on the face of the earth, we see some very interesting trends. One thing that we see is this, that organisms that are found in Australia are much different than organisms that are found anywhere else on the face of the earth. If you look at organisms found in North America, South America, Europe, Asia, those organisms are more similar to one another than organisms found on the continent of Australia. So, interestingly, what we see is that if we look back at the geologic history, we, of course, one time had one big landmass on the face of the earth, referred to as Pangea, that fractured into the continents, the different land masses that we know today. And so when this occurred, interestingly, Australia left first, and so that would indicate that the animals that are present on Australia were separated from the animals on all the other continents for a much longer period of time, for instance, than animals were separated between Africa and South America. And so it would make sense that those animals in Australia are different as a result of the fact that they have been isolated for millions of additional years relative to the animals found on all the other continents. This is referred to as biogeography. Biogeography is an examination, a study of the organisms that are found on the face of the earth, and that study does clearly indicate the common ancestor theory, and fortunately is consistent with the data that we have available to us from the field of geology. So here we have some interesting pictures of animals that are found on Australia and how different they are. If we look at the mammals, for instance, we see that we have over 200 species of marsupials found in Australia. As you know, marsupials are mammals that raise their young in a little pouch on their body. And so we have koalas and sugar gliders and wombats and kangaroos in Australia. In North America, we have one species of marsupial, interestingly, the possum. So mammals are much different in Australia than the mammals you find anywhere else on the face of the earth. We have odd reptiles shown up there at the top of the screen, the fish in Australia. Those are all rainbow fish, a family of fish found in Australia, found nowhere else on the planet. So the take-home message is that animals found in Australia are much more different than animals we find anywhere else. And this is consistent with our study of geology and supportive of the common ancestor theory. We make our way next to homologous structures. And so homologous structures are this. If we look at this picture from our text, we have the forelimb or the arm of a human. We have a humerus, a radius, and ulna. We have carpals, which are the wrist bone, metacarpals, which are the hand bone, and phalanges, which are the finger bones. If we look at a chimpanzee, we see very similar bones, which is not surprising. If we look at a lion, we still see similar bones. We see the humerus, the radius, the ulna, the carpals, the metacarpals, and the phalanges. If we look at a sea otter, we see a very similar collection of bones. And even if we look at a bat, the mammal that appears most different than us, we still see a similar pattern of bones. And so why is this? Scientists look at this and they conclude that these similarities are the result of a common ancestor. And so the common ancestor had this general bone pattern, and it gave rise to all these organisms that we see on the screen. This is referred to as homology. Homologous structures are anatomical similarities between organisms as the result of a common ancestor. Interesting, we see that homology also applies to development. As we look at the screen, we see a picture of a snake, a chicken, a possum, a cat, a bat, and a human. And we see two things of interest, of particular interest to us. In the early developmental stages of a human being, we have pharyngeal gill slits and we have a tail. You've noticed probably that you don't have pharyngeal gill slits and you don't have a tail. And so scientists ask the question, why? Why would you go through this process in development of having these structures only to lose them later? The best answer appears to be that these developmental pathways are the result of a common ancestor that we share with all other vertebrates. And therefore, these aspects are retained within that developmental pathway. And so once again, these are homologous structures, similarities in anatomy that are the result of a common ancestor. We also see, on a related note here, we're still calling this number 3, vestigial structures. And so vestigial structures are traits that are not useful in modern species, but are present as a result of the ancestors of that organism. Wings in ostriches are sometimes referred to as vestigial structures. So the ostriches can't fly, but they still possess wings as a result of the fact that they had a winged ancestor. Some snakes have very small protrusions close to on the lower half of their body that are referred to as vestigial legs. Do they have legs? No. Or rather, are these things useful? No. But they're present because of a legged ancestor. You have a coccyx. So you have this fused 4 to 6 vertebrae that are found below your sacrum at the end of your backbone. Does that serve any purpose? No, but what is it? Apparently it's a vestigial tail, a structure that you possess as a result of your relatedness to ancestors that did have tails. Lastly, we look at DNA as evidence for a common ancestor theory. And so as we look across the world of living organisms, what we notice is this, that as we go from humans to monkeys, they look fairly similar anatomically. And if we look at the y-axis here, we're looking at the number of amino acid differences that are found in these organisms. And so there's very few differences between humans and monkeys. There's a greater number of differences between humans and pigs, between humans and ducks and turtles, and across the board. So what we see is this, that as we look at organisms that are closely related, their DNA, and therefore their protein sequence, as shown in this diagram from our text, is very similar. But as we look at organisms that are more distantly related, we have a higher number of differences that are found in their nucleic acid sequences. We also see this in the case of evidence for evolution and DNA. Much of our non-coding DNA, as we talked about back in our PCR chapter, is composed of viruses that have inserted randomly over the course of evolution. So when a virus inserts, it inserts at one spot, in the case of humans, among our 3.2 billion bases. And where it inserts is often random. And so we can look at the insertion spots of these viruses and tell who we are related to. They serve as nice markers. For instance, in the case of cats, the cat genome, like ours, contains a number of inserted viruses, and these viruses have been used to work out the relatedness of these different species of felines. Lastly, and this is most compelling, as we look at evidence for evolution in humans, we can look interestingly at chromosome number 2. And so as we look at our ancestors, if we look down here at the lower left-hand side of our picture, as we look at our ancestors, most of our ancestors had 48 chromosomes. Gorillas, bonobos, chimpanzees all had 48 chromosomes. But interestingly, there was a chromosome fusion that occurred during evolution, and so us, along with Neanderthals and probably Devonians, have 46 chromosomes as opposed to 48. How do we know that? Well, if we look at the right-hand side of our diagram, what we notice is this, that two separate chromosomes that are found in gorillas, bonobos, chimpanzees are exactly the same. And when we say exactly the same, we mean they're nearly identical in 242 million bases. Nearly 242 million bases are the same. We have vestigial centromeres, which, as you recall, are the center of the chromosome, shown here, and vestigial telomeres. So, in other words, this is the centromere. So this was the middle of this chromosome, middle of this chromosome. Here's the middle, here's the middle. And these telomeres, shown in red, are still present in our chromosome number 2, but they existed at the outside of these chromosomes in, for instance, gorillas. So as we look at DNA evidence, it provides a great deal of support for this idea of a common ancestor theory.